In this video, I will explain how predicted values and contrast can be obtained using BugsXLA. The first example is the clinical trial analysed in a previous video. I will show how to obtain predicted means for all levels of the treatment factor. In cell F2, I've entered TRT, the name of the treatment factor, and in the cell beneath, I've entered an asterisk. Here, the asterisk is interpreted by BugsXLA to mean all levels of this factor. The predictions or contrast box must be checked, and then when OK is clicked, this form appears. The range of cells where the predictions are defined is entered. And then you click OK, and then run OpenBugs. Now when the results are imported back, not only do you get the model parameters summarized, but also the predicted means. Note that since no setting was specified for the covariate FEV1 base, it was set to its default value of its mean. If you look at the note in this cell here, it will tell you how all the different types of parameters are treated when they're not giving any settings for the predicted values. BugsXLA provides a posterior plotting tool to help visualize these predicted means. If you select the cell with the label post plot, and then go to the posterior plotting icon, click it, you get this tool. There are various options here to enhance the graph that's produced. So I just replot that bottom there. So you can show the precision, show credible intervals for this. And there are many other settings here as well that you can use. Also, once you exit this tool, you're left with an Excel graph, and you can use the Excel's own graphing features to change the format of this further. Suppose instead we are interested in some contrasts between the treatment means. In a cell underneath this column here, we could use the commands that BugsXLA understands for contrasts, which is a letter C, bracket, and then you can put the two levels, and that would return the contrast between levels B and C of this treatment factor. If you wanted the contrast between B and all the other levels, you could use the asterisk, and indeed if you wanted the contrast between all possible combinations, you could do this. And just to show what this produces, I will run this analysis and show you the output. Not forgetting to expand the range of the predictions, so it now goes from F3 down to F6. So here you can see it's produced these three extra set of predictions, which are contrast in this case. So this was the contrast between B and C. These are contrast between B and all the other levels. And this is all possible pairwise. In this example, we have a small dose response study into the effectiveness of an insecticide. Here we show how to make predictions for a range of covariate values. In the cell here, we use the V notation followed by the starting dose, the final dose, and the number of predictions we want to make across this dose range. This final setting is optional, and if excluded, then 10 predictions will be made across this range. If we look across to the output that's produced on the right hand side here, we see that for this model, which is a binomial model, we see that BugsXLA makes the predictions in both predicted probability and also predicted odds. If we want to visualize these, as before, we can go to the postplot cell, select that, go to the posterior plotting option, and then these can be plotted. In this case, you probably don't want to show the evaluation points, and you can smooth these, so you replot that. And it's possible to add the data on here. And in this case, the data, which are the red circles, are the crude estimated probability values. In this final example, we have a meta-analysis of clinical trials in which the response is binomial. And what I will show here is how to make predictions for a future draw from a random effect. So in this case, we have a model in which study, as well as the study by treatment interaction, is or are random effects. And what is quite common for a Bayesian analysis of meta-analysis like this is to use it to derive a prior distribution for a future study. So to specify that, we can use this set of predictions here. So here we have the contrast between the treatment and the control level of the treatment factor. And we use the symbol, which is the tilde symbol under the study, to say that we want to make a prediction for a future study, assuming it's come from the same distribution as the studies in this model. Now, because we're going to use this to derive a prior distribution for a future study, we need the results to be displayed back on the link scale also. And this is because by default all the predictions are displayed on the natural scale, but we need it on the link scale because that is the way the parameter enters the model. If we look across at the output, we see that you get the predicted odds for a new study here, and here is the predictions on the logit scale, which is what is known as a meta-analytic predictive prior. If we just go back to the predictions form here, if you click on the help and move to the prediction range, you will see here the notes on all the valid entries in the predictions and how they work.